everybody can see it.
Yeah, it's fine. Yeah.
It's fine. So let's, I guess, we'll try to get a pointer here. Yeah. Okay. So hello, everybody. And I want to tell you a little bit about what we have done in this lab here in the last 14 years, actually, studying bacteriophage viruses. And so the first half of the talk will be a little bit of a history of what we've done in the group, and the second half is really new stuff. Most of it still unpublished, some of it just got published recently. And you'll see how everything is connected to this one theme that we are trying to look at, to research in our lab. So I'm going to talk about bacteriophages, and they've been discovered in about 100 years ago. And they've been called bacteriophages, but these are actually both bacterial and archaeal phages, although it was not known that they also infect archaea. And you can see different types and different shapes of these bacteriophages here. What is common is that they all have some type of a genome. It could be single-stranded RNA or DNA, or double-stranded RNA and double-stranded DNA. I mark a few of these bacteriophages we have been working in our group. So for example, these are T7 images taken by Gili Abramov from my lab. They belong to the Podoviridae family. So they see that they have a large icosahedral head. This is the capsid protein. The DNA is in the middle. And they have a short tail that attaches to the lipopolysaccharides on the E. coli cells when it infects the bacteria. And actually, upon infection, it lyses the cells and then goes out as a new virus. Then we have this tiny Leviviridae viruses. They're about 28 nanometers in diameter. You can see them here. This is an image taken in our lab by my student Or Ruski. And to this family, you have MS2 and GA and AP205, which has been studied by the Pintacuda lab and all sorts of other small, and they are the smallest single-stranded RNA viruses known to man, known to humankind, actually. But I'm not going to talk about this Leviviridae studies, and I leave it to my student to talk whenever he has an opportunity to tell you about these systems. I'm going to rather focus about the family of filamentous viruses, and there you can find PF1 and FT, M13. And these studies have actually been pioneered by the group of Opela down in the 70s and 80s using static aligned NMR methods. And then I started working on PF1 in the McDermott lab, which are very highly ordered class 2 filamentous phage. And in our group, we work on class 1 filamentous phage, which are FT, M13, IKE, and other types of viruses. And you can see how they look like. They have a very long persistent length. It's a little bit like a spaghetti, which would put in the pot for just 30, 40 seconds and then taken out. Some other members of the family have much smaller persistent length and high coiling, but these have very small coiling. Basically, they are made, you can see it here. You have a circular single-stranded DNA, which is wrapped by these P8 coat protein. And the DNA and the coat protein together make up about 98% of the mass of the bacteriophage. They have some minor coat protein on the edges. So this is gene P3 or gene 3 protein, which is required for infection. And you have P7 and P9, which are required for assembly. So in general, this is the life cycle of the filamentous phage, and we're really interested in the different structure that this virus takes in different parts of his life. And so our main interest started actually with these intact phages, which I will talk about first. But then when they infect the cell, they attach to F. pilus, and there was this fantastic cryo-EM map structure of the F. pilus protein showing lipids in the interior of this hollow protein oligomer, allowing the DNA to actually be injected, go in, and enter the cell as a single-stranded DNA. And this single-stranded DNA is replicated, it becomes a double-stranded DNA, or what we call a replicated form DNA. And then expression of the different proteins start. And when this expression starts, you have 11 different genes that are being expressed. And one of them is this gene 5 protein, you can see it here, which actually appears as dimers or homodimers in the cell. And when the concentration is sufficiently high, then this gene 5 will attach to the single-stranded DNA, what is called the rolling circle mechanism, and take this single-stranded DNA to the cell membrane to assemble a new virus. These P8 proteins are actually in the membrane, and they attach to the DNA, and the gene 5 just goes back to the cell, and this process repeats itself. So if the first part is about these intact filamentous viruses, then the second part is going to talk about these premature viruses. And both didn't really have structures when we started working on these systems. And it's really, really interesting because there are differences in terms of structure and dynamics of both these systems, although both actually bind the same single-stranded DNA. So here's a little look on the class 1 phages, and we're going to talk mainly about 2 wild-type phages, FD and M13. And you can see that the only difference is a change in the charge on the surface. Aspartate is replaced with asparginine, and the rest of the phage is actually the same. FD has been studied by fiber diffraction before we started working on it. M13 has not really been studied, and FD Y21 and mutant turned out to be a fantastic candidate for all the different alignment techniques. And the reason you can see here, so if you take the wild type, you get fanning out of the fiber diffraction layer lines, probably because the alignment is not so good. And if you do the mutation, then you get very narrow lines. You can say the same thing from the studies by Opella. You can see the wild type. You should expect 2 lines of N15 because of the 2 leucines. You see broad lines for the wild type. You see very narrow lines for the mutant. But the problem is that the mutation sits in the heart of the hydrophobic interaction core of the virus, as we will see as we go on. And therefore, you can ask the question whether the structure that was told for the Y21 and mutant is actually the structure we're after. So we headed on to look at these FD and M13 viruses in our lab. This is just another example how they generate these beautiful liquid crystals in the cholesteric phase, but we'll just skip that for now. So the first thing we did is we prepared these samples uniformly labeled. You can see these M13 carbon-carbon spectra on a 14 Tesla magnet. Everything was assigned by Gili and Omri. One took the M13, one took the FD, which turned out to be exactly the same, or almost exactly the same. And using this type of data, at least you can say that the particle itself is highly ordered, which was something that was kind of in a debate in the literature. So there were reports that the actual capsid has some polymorphism in it, but we have one peak for each amino acids, and we can predict that we have a helix and we have a small N terminus, which is non-helical, which is expected for these structures. And of course, the next stage was to try and solve the structure of this entire protein. But just to show you what happens with this mutation, so if we measure FD and M13, then you see here in the alanine region that they exactly overlay. There are no changes at all. Actually, if you look at the chemical shift perturbations between the 2 nuclei, between the 2 viruses, and you just ignore this mutation in the aspartate, then the RMSD is about 0.1 or 0.2 ppm, which is like just statistics on the single protein. But then if you take the mutant here in magenta, then you see that you start to get chemical sheet perturbations and changes, which seem a little small, but actually they're significant if you look at how similar they are here. So FDM-13 are almost identical on the atomic level, but Y21M apparently has some structural differences. And Y21M was already solved by all these different techniques like fiber diffraction and static NMR. So we went on to solve the structure of FD and M13 bacteriophages. In our case, it was M13, but it didn't really matter so much. So I won't go into detail because I talked about this in previous years. This is the final structure, which was done in collaboration with Nick Gourakis, which was just moving from the baker's lab to his own lab. And what we see is really this non-helical N-terminal, the helical shape of the coat protein. This is the P8 coat protein. We get a diameter of about 6 to 7 nanometers. We have about 70 angstrom length of the coat protein, and we have this curved helix here. And when you look at the arrangement of the different coat proteins, then if you just take this single nitrogen of the proline amino acids, we see pentamers, which are layered in an approximate C2 tilt axis. It's actually 36.4 degrees, which gives it its slight curvature, and it actually the reason why it doesn't align very well. And the rise is about 16.6 angstrom. So here you can see a single capsid and it's drawn to scale. So this was the structure of the M13, which we managed to get 7 years ago in 2015. And it was a work of a very talented graduate student of mine, Amir Morad. And we were really, really happy. And actually, when we compare the structure to the mutant, what we saw is something really interesting. So if you overlay a single capsid protein, they're quite similar. I mean, the RMSD is not very different. It's a very small value, but you can see some changes in the side chains and slight changes in the structure. But then if you try to overlay a pentamer, then the backbone RMSD increases. And another thing that happens is that the rise changes. And the rise goes from 16.16 in the Y21M to 16.6 in the wild type. Now, this seems like a very small number, but you have to consider that there are about 350 such pentamers in a particle. So if you multiply 350 by 0.45 angstrom, then you get quite a difference in the total length of the particle. And the slight change of tilt to 36.4 versus 36 actually explained also these enhanced curvature and also the difference in the creation of liquid crystals and the impossibility for alignment. So actually this single hydrophobic mutant for the phage change something in the arrangement of these intact viruses as we see them by NMR. Now, when you look at structural sequence alignment of these class 1 phages, then we wanted to see what type of interactions actually dominate the structure. If you look here on the left, then you see that many of these aromatic residues actually stack. And if we count how many contacts we have per residue, you see all these aromatics pop up. So they have very, very strong hydrophobic stacking. You can see it. These dashed lines are actually contacts that we measured in the correlation spectrum. And if we align them, and even with open reading frames found in different types of bacteria, then you see that they have many, many common residues which are preserved between members of the families. And so then we wanted to see if these type of structural arrangement is actually preserved, even if the sequence changes and those key residues remain. So we picked up this IKE phage because you can see that here he has a single arginine as one of the 4 positive charge residues 3 of which actually interact with the DNA. And this is nice because if arginine is unique, then you can look directly at the arginine and measure protein DNA interactions and dynamics and all sorts of things, because there is no, there's no problem with spectral dispersion. So with this IKE project, we actually reverted to cryo-EM. This was done in a collaboration with Ye Chang from Tsinghua University. And what you see is that the overall dimensions are very similar. So again, 6.5 nanometers diameter, about 16.5 angstrom is the rise. The tilt is 38.5. This time it's actually bigger than the 36 that we encountered before. And the nice thing about this cryo-EM structure, you see how nice the resolution is. So these are the actual density maps that but they received for the structure. And the next step was just to compare these two systems and see if they actually have the same binding epitopes. So we can align the sequences of F13 and IKE by their tryptophans and phenylalanines. And what you can see, for example, are stacking of tryptophan 21, 29 with tyrosine 9 in IKE. This is this arrow here. And this is the same stacking as we see in M13 between the tryptophan and the proline, which are these two residues here. So it's exactly the two parts of the capsid proteins. And this already tells you why the rise is going to be exactly the same, because of the same dominant interactions. You can see the same thing in the other part, in the C-terminal part of the coat protein. You have tryptophan with a phenylalanine. Here it's number 45, and here you have the same interaction with the same orientation, but this time it's with the phenylalanine 42. So again, you see the same type of interactions and the same type of binding in both viruses. Although they have sequence similarity, but they also have many different differences in their in the sequence, including the length, which is different in 3 amino acids. These are some more pictures from the cryo-EM, and then the interesting thing is you can actually see the DNA inside. And I think this is the first time the single-stranded DNA is directly detected. And if you look at the contour maps, what you see are these two strands of DNA showing you actually proving that the single-stranded DNA is both circular and both wraps around itself, a little bit like double-stranded DNA, because if you take different sections on the DNA axis, then you see these shape rotates and changes the intensity. That tells you that you have some kind of a winding of the DNA inside, and we could determine the average pitch of the DNA and the handliness of the DNA. Another interesting thing is that you can look at the distances from the different positively charged residues to the DNA in the middle, or at least to see the diameter in which they are. And you see that this arginine is actually the closest to the DNA. So the next thing we wanted to do is actually look at this arginine and properties of the arginine. And what interested us mostly was dynamics of the residues that interact with the DNA. And the reason being that there were prior reports also from the McDermott group about actually dynamic interactions between the DNA and the coat protein. And that tells you that the interactions are not very strong. And it actually makes sense, because then the DNA can slide and go into the bacteria upon infection. So the first thing we do was actually look at the capsid dynamics. We adopted this RNCSA technique developed by the Polanova lab. Only we extended it to 3-dimensional experiments. One of them is CCC and the other one is NNC. This is just an example of N15CSA on an NC correlation experiment. We designed an automated program that picks the peaks and actually extract all these line shapes outside directly. So you get all these blue spectra in a single file that you see here. These are the experimental results. And then automatically it fits the CSA line shapes to all these different blue spectra. And it gives you out an output, which is the CSA and the chi-square fitting, telling you about the goodness of the fit. So summarizing all these different experiments, what we get to see, you can see on the right side here. So these are the CSA of the nitrogen and then the carbonyls, and you see a very, very rigid backbone and a dynamic N-terminus, which is not very surprising because this is really exposed to the surface. But the helical capsid itself is highly rigid and very strongly attached to itself due to these very tight hydrophobic interactions. So then we use the same technique to look actually at the arginine dynamics. And the first thing we do is we took solid arginine, a solid powder, to check that we're actually in the rigid limit. That what you see is in two different temperatures, that the line shapes doesn't change of these N epsilon atom, the arginine. And then the other thing we did is just do the same thing on the arginine. And what you see is a reduced line shape, telling you that the arginine in IKE is actually dynamic. And what we think happens is that there are no direct electrostatic interactions, but these are rather water-mediated polar interactions between the positively charged residues and the phosphate negative charges on the DNA. And this fits very nicely with what was observed from PF1 from dipolar order parameter measurements. So this is the story of the intact full phages. And what I want to talk to you about next is actually the premature form of the virus, which is an entirely different story because gene 5P has to bind and unbind the DNA. So it must be much less tightly bound to the protein itself, to the DNA itself. So what is this gene 5P? So it's a single-stranded DNA binding protein. It's a non-specific DNA binding protein, but it can actually also bind different types of genomes like RNA and double-stranded DNA and so on. In solution or in solid phase, it comes as a homodimer, which you can see here. This is the exostructure sold already in 94. You can see one of the monomers here. You have 4 different loops, which are called the DNA binding loop, the dyad loop, the complex loop, and actually what we now call the core loop, which you'll see why is important, and was kind of ignored many, many years. You can see the dimer arrangement here from the N-terminus to the C-terminus of one monomer and the other monomer. Now there are many, many studies of these free-form double-stranded DNA. You see it's many, many years ago. And actually studies of the binding to very small oligonucleotides. So this is one of these solution NMR studies where the DNA was labeled with some paramagnetic species. And if you look at the relaxation time of the protons of the different residues, you see in every place the black tells you the reduction in the relaxation times. And you can see that the DNA binding loop indeed binds DNA because of all these reduced relaxation times. You can see some in the dyad loop and also some here in the C-terminus. So the DNA actually binds in this region, going down. There was an attempt to model the structure with the DNA, but there's no structure of the protein bound to this entire single-stranded DNA protein. And in fact, when you do this experiment, you have to bind 2,000 proteins to about 8.2 kilobase pairs of DNA. So this is our attempt on this system. What you see here is just an agarose gel with different ratios of nucleotide to proteins. And as we bind more and more in protein, what you see is that the complex migrates less and less until it saturates between 3 and 5 nucleotide per protein. And this is actually the value that was reported in the literature in much, much older studies. It also shows you that actually we get the complex very nicely as reported. The other thing you can do is create mixed samples. So you can take a labeled DNA with a natural abundance gene 5P. This is this blue spectrum. And you can see the ribose sugars very nicely. You can see the thymine methyl groups popping up at their unique chemical shifts. You can see contacts between the thymine and the ribose. We actually see many, many more base signals, but we just, I increased a little bit the contour levels here just to not to confuse the eye. And we can assign this entire single-stranded DNA, at least by residue type. Now, the other thing you can see are on the diagonal signals of the protein, which is in natural abundance in the sample. Now, if you reverse label it, you use a label gene 5 with a natural abundance DNA. This is this orange spectrum. And then you see a very, very nice high resolution solid state NMR spectra with no DNA signals. But you see that that these signals sit exactly where we have the diagonal signals on the non-labeled sample. So these things together tell us that we have a very nicely ordered complex of this gene 5 with the DNA, with the right amount of protein to DNA binding. Now, the next step was to see, try and see how far the DNA is from the protein, and actually to prove by NMR also, not by gel electrophoresis, that the DNA and the proteins are bound. So the easiest experiment to do is just a reader experiment where you look at the phosphorus spectrum and you hit the carbons with the pulses, and you can see the nice reduction in the signal. And then we can draw the reader recoupling curves and fit it to a system with 3 carbons. And this gives you a distance of approximately 5.5 angstrom between the phosphate backbone and 3 carbons on the protein. Now, the choice of 3 is actually a game between a 2-spin system, 3-spin system, 4-spin system, etc. If you take 4 carbons, that doesn't change anymore the shape. But if you take 2 carbons of 1 carbon, then you get unreasonable results and the shape is not correct. So we believe that we have a mixture of between 2 and 3 carbons, and that makes sense because you have many different residues that bind the phosphate. So this is an example of 3 carbons at approximately equidistances to the phosphates. But when you have charged residues interacting, you may have 1 or 2 carbons. So it's probably kind of a mixture, and the next experiment, of course, is going to be 2D carbon-carbon experiment dephased by the phosphates, and then we will know exactly which residues bind the DNA and at what distance. And that will help us in the future to find the structure with the DNA inside, structurally, I mean, experimentally. So that was the direct detection. The next step was to actually assign the bound and the free form. So you see it here. This is the type of data that we get. This is just classic NCACX, NCOCX experiment, 2D experiments, inadequate experiments, and so on. So a lot of work by Smadar from a group who actually worked many, many years to make sure that she can make all these samples, which are really, really complicated. Gene 5 tends to aggregate, and it's difficult to extract a single stranded DNA in proper form. So there was a lot of work and effort done here. And from her data, only an undergraduate in our lab, actually from the Odyssea program, which is a program we have in the university for high school students. So she worked for me for a year and managed to assign the free protein. And the next step is, of course, to compare the free and bound and to see if the structure changes. So we expect small changes, but this came as a big surprise. So if you look at the overlay, then you see really very, very big changes. At the first sight, I thought, okay, the spectra are biased. It looks like the magenta spectrum is a little bit unreferenced, but this is actually not correct. It's referenced correctly. And you can now pin down all these different chemical shift perturbations. You can see really significant changes across the protein, and we can draw them here. The RMSD per amino acid, you see it goes up to 4.3 angstrom even if you scale down the N15 shift by a factor of 4. And you can see the major changes in the DNA binding loop, in the C-terminus, somewhat in the dyad loop, and we'll see a little bit more about this later, actually in the next slide. But the changes are really across the entire protein. This is really surprising. So we've done two things. We plotted this chemical shift RMSD for the entire protein, you see by residue number. And also the other thing we did, I took out these so this is actually a quadratic average of the torsion angle. So it's a square root of phi squared plus phi squared. These are the torsion angles predicted from the NMR chemical shifts. And on this, what we do is we plot the moving average, the 5 residue moving average. And what you can see from this plot is regions where you have really significant changes in the orientation and the torsion angles and chemical shift of the protein. So 4 of these places with very large changes are drawn here, the DNA binding loop. Now you see this core connecting loop here, which goes from beta 4 to beta 5. We have big differences in the C-terminus, which are manifested in the RMSDs, less so in the torsion angles. And also in this small loop here, you can see these changes here in the RMSD, in the torsion angles, but less so in the RMSD. So these are marked in different colors. So, but overall, I mean, the RMSD is about 1.3 ppm, which is really large when you compare to supposedly similar structures. So naturally, there's going to be a really, really significant change in the structure, and what we have to do is actually solve the structure. So this is what I'm going to talk about, and this is all the work of a very talented student of mine, Yoav Shamir. And all this data that I showed you that was acquired by Smadar and some data that was acquired by Yoav was used as an input to the Explore NIH program, which failed in approximately the 100 times that we tried to use it in the beginning. But eventually we realized what we have to do. So this is kind of the way that we use the input to explore. So we took distance restraints between 2 to 8 angstrom to each carbon-carbon peak that was picked by the software that Yoav wrote, and then filtered for diagonal peaks and side bands and so on. So all these blue dots were taken into account in the program. Then we realized we must have at least one sparsely labeled data. So we took a 1,3-glycerol sample, and what we did is aggregate the restraints from the uniformly labeled sample and the sparsely labeled in a way that if you have a sparsely labeled restraint, which is less ambiguous than the uniform, then you drop the ambiguities from the uniform labeling and you remain with the sparse labeling. But when you assign this ambiguity and you use sparse labeling, so you have to decide on two things. So first of all, on the error in the fitting in the assignment of the peaks, which we took to be 0.3 ppm, and the number of ambiguous contacts that you use, which we took to be 20. And the last thing is a score for the scrambling. So when we do these 1,3-glycerol labeled samples, then we have a lot of scrambling of the data. And if statistics tells you that there is a chance of more than 40% to see across peaks, so if it has some known scrambling, that we count this in our data. And now the next thing we did is to say, okay, we have a lot of structural changes, but still there is must be some relation to the X-ray structure. So we decided that every distance that violates a 16 angstrom threshold in the X-ray structure, we're going to throw out and therefore reduce the ambiguity. So we assume that the overall shape stays the same. And if our restraint is 8 angstrom, then nothing can be more than 16. So it's a very loose restraint that we take here, but it helps a lot. It's actually crucial for the structure determination. And the last thing is what we have always with the problem of calculating homodimers. So you can always have contacts between the dimers, which are really, really problematic. But again, we have this X-ray homodimer structure. So what we did is looked at all these dimer contacts, and everything that was less than 7 angstrom and it appeared in our data, we just dropped it and we never used it again. And by using calculations on synthetic data, trying to calculate the homodimer, we saw that when you do this, then you get a very accurate structure, and there's no, I mean, the structure doesn't deform because of these homodimer contacts. So we use this, and of course we use the angular strains from Talos. But what one must keep in mind that Talos is really, really good for helical structures, less so for beta sheets. And when you have loops, it's not working very, very well. And actually, there were quite a few violations of Talos we had to drop on the way of calculation. So here's a little bit how it looks like. So the first run of 250 structures, we keep 25 with the lowest energy structure. With the lowest energy, this is this first structure that you see here. So on the blue, you get the C alpha RMSD, and on orange, you get the energy score from Explorer. Then we do a cutoff of 16 angstrom from the X-ray structure and remove violation. And this second run already gives you a much lower energy. Then we start doing cutoffs on our average structure and repeat our calculation of the Explorer structure. And we do this about 11 times in total until we go down to a cutoff of 8.5 angstrom. And what you see is that down to about 10 angstrom, the C alpha RMSD goes down and stays more or less the same, slight drop off. But what does go down is the energy when we do this. And then finally, we do a refinement and an explicit solvent. And if you calculate the RMSD for the entire structure, you get a 1.5 angstrom RMSD for the 10 lowest energy structures. Now you see here a picture of the protein, which I'll show in a second, but all these blue lines are actually the long-range restraints we had in the final run to calculate the structures. And immediately see that your loops are less defined, which is not surprising. And these old solution and MR calculations also have the same problem. The same thing goes to the diode loop, but the central core of the protein is highly defined. This is a very important part of the protein, which I'll talk about in a second. Here is the convergence of the structure. So you see from the initial structure in its contact map all the way down to the ensemble of 10 structures and the contact map. Now you can draw the C alpha RMSD per residue, and then you see a clearer picture. So the DNA binding loop, again, the RMSD is large because we don't have many restraints for this part. And we are sure that once we get these distances from the DNA, this will be much better defined. The other loops also have a somewhat larger RMSD, but then the remaining of the protein looks pretty good. And if you go to all the classic software structure calculation and validation techniques, then for the ordered region, you get an RMSD of 1 angstrom, which is more than valid for these type of calculation. This again is this picture with the long range restraints. The next step is, of course, to compare our structure with the free structure, that you see here on the right. When we do this, mind you, we have an RMSD of 1.5 for the entire protein, and the RMSD between the two structures is really huge. It's 6.3 angstrom. These are really unsimilar. And now there are a few things that we should really notice carefully that happened. So these are the distances between the DNA binding loop and the different other loops. So this is this core loop, which had this huge torsion angle shift, and you can see the big change from here to here. You can look at the distances between the core loop and the diode loop, and the core loop, and the DNA and the core loop, which I didn't draw here. So in order to estimate these distances, I mean, because it's not so well defined, so what you have did is took an RMSD, he took an average of all the distances between the DNA loop and the core loop, and all the distances between the DNA loop and the diode loop. And these are these average differences in angstrom between the different loops. That tells you that the protein actually shrinks. It's like it's trying to capture the DNA. Now, these are numbers, and then you can see in this beautiful plot how this space is being filled. So the green is the complex structure, and the black is the extra structure. We've done the same thing with the solution NMR. So the deviations are not that clear, but again, what we have is filling of this cleft here where the DNA binds. So this is actually a manifestation of these numbers in this picture that we've seen before. So overall, what happens is that the protein actually reduces a little bit its radius of duration. It shrinks a little bit, and it just brings all the different loops closer to the DNA so it can bind the DNA. You can see that, so this is kind of the line of the X-ray structure, and this is kind of the line that defines the complex structure, the DNA in the complex structure. Now, finally, what you can see is how these structures change. So this is just a low energy software that gives you some kind of a visualization of what happens to the structure. And what you see are these two parts getting much, much closer together. And mind you that we have many, many different correlations that ensure that this actually happens. And you can see what happened to the N terminus that shifted all over here and the C terminus that shifted all over here. Now, although the structure seem to be quite different, if you compare the contact maps, many of the contacts are preserved. So all these contacts in blue are common to both structures. So that preserved its main beta sheets, although they're pretty short, so sometimes they're defined and sometimes they're not. And all these green contacts are what happens in the bound form. And you could see that many, many different regions actually get closer in order to bind the DNA. Yeah, so this is the story. And if I just summarize, then I showed you that we calculated the structure of these intact phages, those M13 and IKE. Currently, we're working on refining the IKE structure with our solid state NMR data, hoping to get a better defined and also the N-terminus part. And the other thing we showed you is the structure calculation of the gene 5P in complex with DNA, which has actually been an open question for more than 30 years, which is now solved. We now have initial restraints to also model the DNA inside. And with some more experiments, once our probe is not broken anymore, we'll be able to measure many more of these phosphorus carbon distances, and hopefully next time I'll be able to show you how we model the DNA in this binding cleft. So I have to thank my group. This is my current group member. This is really work of Yoav and Or, Meital, who taught Roni everything she knows. And when she came here, she was like 16 years old. And Yaron, who just finished in master on a different topic. These are former students who worked on this project. And of course, I have to thank the funding agencies, and with what Bob started, and I want to invite you all to the Rocky Mountain Conference. Copper is beautiful and lovely, and we're coming up with a very, very nice program that will be distributed in the next coming days. And I thank you for your attention.
Okay, well, thank you very much, Amir, for that really in-depth, thorough review of phage coat proteins and DNA binding and so forth. We're a little behind time in the schedule here, so is there a question or two from the audience? So there's one here. Have you tried P41 C13 reador experiments with the sparsely C13 labeled GVP? Also, did you consider F19 labeling for long distance measurements in the GVP assembly?
Well, the PC reader experiments, we have all the samples and the probe is broken. So these are going to happen the moment the probe will work. So yes, we have samples which are sparsely labeled, and we have samples which are fully labeled, and this will happen probably in the next couple of months. And F19 for long distance measurements. Well, that's of course a fantastic idea, and since this comes from Gal in Tatiana's group, so they have all the hardware and equipment to do it. So perhaps it's a call to do it together, because we cannot do it in our lab, but yeah, it's definitely a good idea.
Okay, Kristen Griesinger has a question. I was wondering regarding the filamentous phage EM with the DNA. That you resolve the DNA as a dimer. Does it mean that the symmetry along the phage axis and the DNA are the same? I hope that formulation of the question is understandable.
Yeah, so the formulation is understandable, and my answer for this is as follows. So the pitch of the DNA is probably doesn't have the same symmetry of the capsid. And I'm not an expert in cryo-EM, but this has come up in many, many different discussions with the reviewers of the paper, actually, because the capsid is right-handed and the DNA was actually left-handed. The thing is that the resolution of the DNA is 6 angstrom, and the resolution of the capsid is about 3 angstrom. So I can't give a precise answer. There might be some common pitch after several cycles, but it really calls for much better resolution on the DNA part. Now, it was not minimized using the same symmetry. So the cryo-EM guys, they have techniques to minimize with different symmetries, different parts of the complex, from what I understand. But the problem is that we know that the phosphates are somewhat disordered in the structure. So I'm not even sure. We know that actually from Opella's work, very old work, that it gave always broad lines even in the aligned samples. So I'm not sure this can even be determined precisely. Since the protein is a non-specific single-stranded DNA binding protein, it doesn't care actually about the pitch of the DNA inside. I hope my answer was understandable.
Okay, so I was going to suggest that, if you want to get some DNA protein contacts, that you might think about trying to do FS Redor experiments. You're doing Redor, now maybe you were doing FS Redor, but they tend to be quite accurate. The results, down to 1/10 or 2/10 of an angstrom if you have good signal and noise.
So we plan to do all these experiments. They're all ready, but our 4mm probe is broken. Okay. So hopefully they'll be done. These are all good ideas and all planned, and hopefully they.
Okay. All right. So if you could stop sharing your screen for a moment, Amir.
Yeah. Sure.
Yeah. And the next speaker is Enrico Rivera, who is from Florence, University of Florence. And Enrico was an undergraduate in Florence. He did his PhD with Claudio Lucanat and Ivano Bertini. He was in a postdoc at the University of Florence, and he spent a little time, 6 months or so, here in Boston, doing some DNP experiments. He then became a staff scientist, assistant professor, and now in 2022, he's an associate professor at the University of Florence. And today, he's going to tell us about solid-state NMR studies of immobilized enzymes. So Enrico.
Can you hear me? Thanks a lot for inviting me, and also I feel a little bit the pressure of being the last speaker of this series.
Well, not the last. Necessarily. Okay.
The finale, at least. Let me share the screen, which I cannot find anymore. Here it is.
There you go. All right. So we can see your.
Yeah, I think you're seeing all my presentation from the end to the beginning, but. Now it should be in the right way. So again, thanks.
Okay, Enrico, there's a overlap of maybe one of your slides on the right-hand side?
I don't know what exactly you are seeing. You should be seeing.
And now it's better, but it's still a little bit there. But why don't you go ahead? Okay, it's okay. Right?
So, just a quick glance of the place I'm coming from. This is the Magnetic Resonance Center of the University of Florence. The numbers that you see there, the magnetic fields expressed in proton-Larmor frequency. And this is just to recall that we are open to access also to solid-state NMR users through a series of access schemes, including the recently founded Panacea. So for anyone who is interested in accessing our instrumentation, you can refer to our website and see what are the prospects for accessing instrumentation. I decided to give to this talk, say, a literary cut, just to reflect the fact that I have a little bit of humanities background. And I selected a few stanzas from a poem by Kostas Kavafi, who was a Greek poet in the beginning of the last century. And this poem, Ithaki, just says, begins with, as you set out for Ithaca, hope your road is a long one, full of adventures and full of discovery. And I think this reflects quite much the idea that at least I have of research. Actually, this also brings the idea of Ulysses, the Homeric hero, who on his way home made a lot of discoveries and experiences and so on. And in the Middle Ages, since the Odyssey was not known, at least not translated into Latin, there was an alternative tradition that said that Ulysses and his fellows died by crossing the columns of Hercules. And this is the Divine Comedy. This is just to recall that the pursuit of knowledge is not always easy, because in the end, Ulysses ends up in one of the lowest of the bulges of hell. So perhaps it's not the best choice ever, but okay, let me try to follow on this idea. I try to give a little bit of motivation to the research that we are doing, but you will see that as for Ulysses, the motivation soon becomes different. So in general, I'm interested in characterizing the interface that is between proteins and materials, because this pertains a lot of fields, environmental, photochemistry, formulation of vaccines, catalysis, biomineralization, or whatever you can think of. So there is a lot of chemistry going on at the interface between a protein and a material. And for a number of reasons, the way to approach these systems is NMR. Because NMR can reveal the properties of the protein in terms of structure, dynamics, reactivity. It can inform about the morphology and the properties of the material. It can also inform about the nature of interactions. What is covalent or electrostatic, or how much the surface or the protein remain accessible. So NMR is full of information on this kind of systems. And in particular, we have worked for quite a while, as Bob was recalling, on enzymes embedded in nascent inorganic matrices. So the inorganic matrix is templated by the presence itself of the protein. And of course, as you all know, NMR can inform you about the state of the protein, because protein usually gives very well-resolved peaks and maps that you can interpret in terms of contact, as you've seen brilliantly in the presentation by Amir. And also, at the same time, NMR informs about the state of the material if the material has active nuclei. And for instance, silicon has an active isotope, so we are happy. The problem is that when working with this kind of systems, you run into a little bit of problem with sensitivity, because you're happy when you're doing materials NMR because your rotor is full of the material that you're studying, and so you have a lot of signal. You're happy when you're working with pure protein systems because besides a little bit of solvent that is needed to preserve the mobility of the protein, then you have a lot of protein in your rotor, roughly. We are talking about 70 to 50%, so quite a lot. Now, if you work on composites, depending on the nature of the composite itself, you have a problem of sensitivity because the protein is few, the material is also reduced, so whatever you decide to look at, you're probably doomed in terms of sensitivity. For this reason, as Bob recalled, we went for DNP, and at the time, we are talking about 2014, so 8 years ago, I'm growing old. We had a decent enhancement at 700 using the standard DNP juice on the enzymes that we were interested in, which are superoxide dismutase. Here, I don't see the mouse. Oh, here it is. That is up here, or cobalt the catalytic domain of MMP12. But also, and this is approximately where the new story begins, we were able to observe the resonances of the scaffold molecule, which is a small polycationic protein that is lysozyme. And now, so the question becomes, so the motivation changes. It becomes from the characterization of the material. The question becomes, what about the scaffold? What happens to the scaffold? So, what is the role of the protein, and what happens to this protein after the material is formed? So let's start with the role. The role is most probably to drive the precursor molecules, which are negative, and this protein is highly positive. With a little bit of difficulty, and it was not so easy to achieve, we were able to locate one precursor molecule inside the crystal of lysozyme. That is this small tetrahedral titanium species in a very positively charged patch in the protein. So the protein may reasonably have a role in pulling together the precursors for this synthesis. Now, what happens to the protein after the synthesis is completed? And this is quite less defined. There are two main hypotheses. One dates back to the original publication where this method is presented, that the lysozyme remains folded because it remains active and is sitting somewhere within the composite. And another hypothesis, which is more recent and based on the analysis of Sachs data, is that lysozyme partially unfolds and remains stuck outside of the composite. So there's no protein inside the composite, only outside of the protein. So outside of the composite. Well, we had a look at how the protein is, and in line with what we saw with Bob, but with DNP, the signals of the protein appear to be quite well spread. There is a little bit of increase in the signals of arginines, and this is most likely related to the fact that there are some charge interaction going on to decrease the mobility of arginine side chains. So we can rule out the possibility that lysozyme partially unfolds. Now we need to, but still we are not sure of where is lysozyme. And so we made some tests. This is the the only non-NMR thing that I will show today. We made some models. So the protein is electrostatically trapped inside the material, or it is sterically, mostly sterically trapped inside the material, or there is a charged electrostatic interaction and the protein is outside, as postulated by the latest paper, 2019 paper. And the expected behavior is that if you wash with water, the protein remains there. If you wash with a denaturant, everything is removed. If you wash with a denaturant on low ionic strength, however, the protein will be released only if the interaction is mostly spherical and the protein is inside the material. And finally, if you wash with sodium chloride, so high ionic strength, 6 molar sodium chloride, the protein should be released only if it is an electrostatic interaction outside of the material. So we did the tests, and the behavior is that, of course, the protein is retained in water. It's released upon denaturation on ionic strength. It's released upon denaturation on low ionic strength, which seem to point towards a steric interaction. And it's only 20% is released, meaning that most of the protein, at least 80% of the protein, is sitting inside the material. So to cut this very long story short, hypothesis 2, where the protein unfolds and sticks outside of the composite, is basically disproved. So we can say that hypothesis 1 is more or less correct. Okay, but where is the protein? So again, the question changes. It's what is the fate of the protein, and where the protein actually ends? And again, NMR can provide an answer, provided that you can see a carbon-silicon correlation, which turns out to be quite challenging because we are working on natural abundance. So you have 1% on carbon, 4% on the silicon. So the possibility of having a contact is 4 out of 10,000, which is not very much. And the two frequencies are very close in frequency, so it's also challenging in terms of hardware. That cannot be done with a standard hardware. So we decided to pass through a shortcut, which is protons. Again, I have to stress that the fact that we are sensitivity limited, and this is where we had another detour. This is how the spectra look like. The signal to noise ratio is between 5 and 10 for silicon and carbon respectively. We decided to give it a try on multivariate analysis based on the idea that we can separate the signal from the noise, because the noise will not have time dependence, whereas the signal will. And so the indirect evolution gives us a way to filter out what is signal and what is noise. And it turns out that the multivariate curve resolution is our low-cost alternative to DNP, because you only need a PC to do it. And this is the difference between before and after application of the denoising, of this denoising approach. So starting from a signal to noise ratio of 11 and 5, we end up with a signal to noise ratio that is around 40 and 20 for carbon and silicon respectively. On this basis, it's really, it becomes easy to extract slices of the proton dimension along the different axes of the spectra. And what we could see is that, and this is, I will spend some time on this slide because it's it condenses almost 5 years of work. So it's quite a lot of information for a single slide with three panels. The proton silicon head core spectrum, so the silica matrix, is receiving magnetization from 3 different proton sources. 2 are clearly attributable to either silanols on the surface of the protein, so the peak to the upfill peak, and to absorb water. that is in the peak in the middle, and one part which comes from hydrogen bonded stuff. We refrained from speculating in our paper, which is submitted and hopefully under minor revisions, we refrain from speculating that this is actually a crosstalk to the protein, but there is some quite a lot of identity between the spectrum that you get projecting the proton dimension of the silicon spectra to what you get by projecting the proton dimensions in the carbon spectra. And in the carbon spectra, you can always trace it to the protons of the protein, which are shown in black for the sake of clarity. And you see the aliphatics, you see the C alphas, you see the water, and you see the NHs. And if you wash this composite with D2O, the peaks tend to shrink, and the identity, the overlap between the peak at 7.5 around 7.5 ppm becomes quite apparent. So in terms of line width and in terms of chemical shift. So even if we do not want to claim that it's the NHs that are providing directly contact to the protein, from the protein to the silicon, in any case, in the presence of the protein, there is a network of hydrogen bonds that is lost upon protein removal. So as long as the protein is there, there is a network of hydrogen bonding contacts that are involving both the protein and the silica. When the protein is removed, this network of hydrogen bonding is completely lost. And this peak, the 7.5 ppm peak that is in this picture is not present in the spectrum. So just to give a conclusion, and I didn't want to take too much time, so I tried to be as concise as possible. So lysozyme remains in contact with the surface of the silica. This contact is quite intimate, but it's not at all a covalent interaction as observed for other systems. The interactions do not alter the fold of lysozyme. Lysozyme remains folded as it is folded when it's free in solution. So we can dismiss the hypothesis that lysozyme is disrupted by the interaction with the nascent material. And since denaturation, but not high electrostatic, high ionic strength is necessary to remove the protein from the composite, we can conclude that lysozyme is sterically trapped within the silica. Back to the poem by Cavati. The last stanza of this poem reads that if you find Ithaca poor, so maybe if you publish in a low impact factor journal, Ithaca won't have fooled you, because wise you will have become, so full of experience, you will have understood by then what Ithaca means. So it's not the goal of finding out where lysozyme was, but rather the development of the methods that we followed. So from the DNP with Bob to the mathematical denoising with Francesco, who is sitting here in the audience, is what led us in this research. And before going to the acknowledgements, I would like to remember Shimon, who was already recalled in this series, because for a number of reasons, both scientific and personal, if it was not for him, this research wouldn't have ever been carried out. So with this recollection, I go to the real acknowledgements. This work started with Claudio Lucinat and Giacomo and Marco, Vito and Linda back in the days. Now it's carried out mostly in collaboration with Lucia, my postdoc, who is going to leave quite soon. At the end of the month, she will not be my postdoc anymore, and I'm sad. Francesco, graduate student, and some colleagues in the chemistry department, Emiliano and Giovanni, and longtime friends that I also see in the audience, and I'm very happy for this, that are Vlad and Gil. And I thank you for your attention while inviting you to come to the Chianti workshop.
Okay, thank you for a very nice talk. And that's the famous Chianti logo up there at the top that you see. All right. The people sitting on an energy level. Uh, so have you done these experiments I mean, you're spinning at what, 12, 15 kilohertz with most of the stuff now still? Or have you gone up to 100 kilohertz or 60 kilohertz?
No, thanks for the question. I can close the presentation. I don't think I need it. One reason why we didn't go to high frequencies is because the rotors are expensive. No, sorry, I meant that one reason why we didn't go to high frequencies is that we are looking for weak interactions. And when you spin faster, the averaging of magic angle spinning will become more dramatic. And so if we have a little bit of spin diffusion that is assisting us in seeing correlations, then this small assistance will be lost upon high spinning. Of course, we will try. We will also try high field, but for us, it was very important to prove that we could work with low frequency, low spinning, because of the possibility of using recoupling.
Yeah. Okay. All right. Well, I mean, you're trading off resolution for maybe some ability to get distance information, but if you can, I mean, if you do get resolution in the proton spectrum, for example, you can do something like an RFDR or a BAS type recoupling sequence and use the protons. Anyway, that's yeah. I mean, you know, I've always thought those systems are homogeneously broadened. And so if by going to high field, which is maybe more important than spinning fast, you'll probably increase the resolution significantly. So are there other questions? Kong, do you have some questions?
Oh, just a quick question. I mean, like, I have a collaborator here who also wants to do something similar, like 29 silicon and carbon. So I don't know, like, is it very is expensive to label the carbon 13 or 29 silicon? Is it more from the cost perspective or some other reasons that you don't label them?
Well, the answer is labeling the silicon is quite, it might be cheap. Labeling carbon on this protein is a hell of a mess because this protein is not expressed in bacteria because it has bactericidal activity. So if you try to express it in bacteria, it kills the bacteria. So one way that we can label the protein, and this is something that we are currently working on, is to do reductive methylation of the lysines. Since it has 21 lysines on the surface, you add 13C label formaldehyde, sodium borohydride, and it labels with carbon 13 all the lysines, which are very much reactive. And this has been published. There are a couple of papers both in NMR and in X-ray. So this is the way we will go. And for this, we will go to high spinning frequencies because we have to record proton-carbon correlations in the methyl region, which has, of course, poor resolution if you don't go to fast spinning.
So you can label lysozyme, Enrico. So there's a bacterium. It's a bacterium that occurs in bathrooms when you don't clean them properly, this black stuff. I've forgotten what the name of it is, something Niger. But anyway, so Chris Dobson's group at Oxford actually did label it, and I can send you some papers on that.
I know the papers, and I think is a very good made some very good results with solution NMR on this. So I think we will do that strategy.
Okay, so Gil Gobus asked you a question here. Can you we try filtering broad proton lines and try to look for narrow proton lines in the proteins like methyls, extra information from them. Perhaps even try for proton detected 2D spectra starting with either carbon or N14.
Yes, thanks, Gil, for the suggestion. Actually, as you recall, we discussed it now 3 years ago when I was back there in Israel. Yes, it would be a good idea. Actually, I had started trying the nitrogen 14 filtering. Unfortunately, the instrument is down for the moment, but it will be back up soon. So when this happens, I will recover the nitrogen 14 stuff.
Okay, all right. So, okay, if you put your Chianti slide back up, can you put that back up? Yeah, we'll just close the meeting. We don't have any more. Yeah, so as I said at the beginning of this, this is the finale, the grand finale of the ZOOMinar series, at least for the last couple of years. And the reason for this is that there are 5 in-person meetings scheduled. And the first one is the one that you've seen advertised right here, the Chianti Workshop. It will occur June 19th to 24. And following that will be the Euromar meeting, which is July 10 to 14 in Utrecht, the Netherlands. And then Amir has previously advertised the Rocky Mountain Conference, which will be at Copper Mountain, Colorado, also in July 24 to 29. So if you're going to Copper Mountain, I suggest you take some oxygen bottles along. The base elevation there is 9,500 feet. So you strain a little bit to breathe when you first arrive. At the end of August, the International Conference on Magnetic Resonance and Biological Systems will be held here in Boston, August 21 to 25. And finally, the Alpine Chamonix meeting will be held September 4 to 9. So there are 5 in-person meetings for everybody to attend. And that sort of obviates the need for these virtual seminars we've been having for the last almost 3 years now. So I think I owe, in particular, a giant thank you to Kong, who's really arranged these things. He's done 99.9% of the work and deserves whatever credit or blame for the success of this series. And we do intend to maybe restart them in September, but for the months of June, July, and August, we're going to take a vacation. So anything else, Kong, you would like to say? All right.
No. I mean, like, thank you for for taking this like crazy idea that we had like when we started it 2 years ago. I mean, it was just done over, I don't know, just 2 or 3 days. Then we started it, and thanks for doing. I mean, like, yeah, supporting my crazy idea, and it has been going on for 2 years. And yeah, let's see if we're gonna continue this in September, and then we will discuss the line of the speakers again, and see you tonight.
Yeah, everybody stay tuned. You will get an email when the series restarts.
Okay.
Email or 2 emails, okay? And so thanks again, Amir and Enrico, and we hope to see you at one of these meetings sometime soon, okay? Bye.
Bye.